So in principle, the background of the introduction of these new chapters by the United States Pharmacopeia is actually that there's an older chapter, which is called Chapter 231, which has been implemented roughly 100 years ago, and studies have proven that it's actually not fit for purpose because it's a very old test based on wet chemistry, and the results don't really match what is actually in your sample. Therefore, United States Pharmacopeia actually decided to redo this chapter, and the outcome were two additional chapters, 232, which actually sets limits for different elemental impurities being present in drug products, and chapter 232 that actually describes two analytical methods to actually analyze for elemental impurities. And one of them is being ICP-OES, which is explicitly mentioned in that chapter. The other one is ICP-MS. Of course, also alternative procedures or methodologies can be used to analyze elemental impurities, and Chapter 233 also explicitly states the validation criteria for these alternative methods. In principle, if you look at the two techniques which I mentioned, ICP-OES and ICP-MS, they are both well capable of doing the job. So both have enough detection sensitivity and dynamic range, but especially if you look at drug products with a very high daily dose, that means the amount that you have to detect in your sample becomes smaller and smaller. And therefore, with ICP-OES, you might come to a certain threshold limit where your limits of detection that you can actually achieve with the instrument are very close to what you need to achieve. ICP-MS, on the other hand, has a higher detection sensitivity and can therefore go lower. And this would be, in my recommendation, it would be the more future-proof choice if you want to select an instrument to go for. It's basically all about validation of the method. I think this is the primary concern of the customers that are faced with the analysis of elemental impurities now using atomic spectrometry techniques. So what we can help in order to achieve this goal is actually to provide methods, to provide tools to actually almost automatically validate their methods. And of course, with our Cutegra software, we have a very intelligent tool set. The Cutegra software also offers you a full suite of quality control tests and a dedicated tool for actually reporting your results. That means you can generate tables specific to your needs. You can, for example, include information about the origin of a file, so you can actually monitor when a file, a data lab book, was created, who created it, who acquired it, and who made the last change to it. Of course, it also supports electronic signatures in order to assure the originality of the data. The reporting tool can actually do much more in order to help you to validate your method. So you can actually generate tables specifically reporting a subset of samples of a larger batch. So you can actually pick, for example, samples corresponding to one drug product and report them in a dedicated table and automatically display, for example, the recovery that you achieve for one of these spike tests, and automatically include as well the relative standard deviation, which is one of the requirements for validation as per USP Chapter 233.